While doing research on the cell cycle, we discovered that there was a remarkable similarity between it and the cycle of human relationships.
The cell cycle starts with G1, where the decision is made to start the process of cell division. This is similar to a person deciding that they are ready to start looking for a partner.
Synthesis is the next step. This is when the cell copies all the chromosomes. Following that is G2, when preparations are made for the cell division.
Mitosis is the more simplistic of the two cell divisions. We compared it to the more old-fashioned type of relationships. In prophase, chromosomes start to condense into discrete chromosomes. The nucleoli disappears and the nuclear membrane breaks down.
By the end of prophase, the centrioles have migrated to the opposite poles. The chromosomes are moving to the center of the cell, and the mitotic spindles start to appear. This is comparable to a classical scene when people pair off and slow dance, like chromosomes in the nucleus.
In metaphase, spindles form and the chromosomes take up position on the metaphase plate in the middle of the cell. Each double-stranded chromosome is attached to the spindle tubule at its centromere. This is like when two people come together to form a couple.
In anaphase, centromeres of each chromosome separate. Each sister chromatid now has its own centromere, and is called a chromosome. Spindle microtubules attached to the centromere cause the chromosomes to move. This is similar to a couple having to separate due to travel.
In telophase, the nuclear membrane begins to reform around the unwinding chromosomes, and the nucleoli begin to reappear. The two daughter nuclei will be identical to the nucleus of the parent cell.
Cytokinesis accompanies telophase, but is not actually technically part of mitosis. It is when the cytoplasm divides. The results of the entire cell cycle are 2 new identical daughter cells, both with the same number of chromosomes as the parent cell and half the organelles from the parent cell.
The importance of mitosis is to replace old or damaged tissues in the organism with newly created diploid cells. Meiosis is the second type of cell division used by cells involved in sexual reproduction. It's so much more sexual that we likened it to the relationships found in Jersey Shore.
In prophase 1, the nuclear envelope breaks. DNA is exchanged between homologous chromosomes in a process called homologous recombination. This results in crossover. This is important for genetic variation and combination of new alleles. These paired and replicated chromosomes are called tetrads. This is like the Jersey Shore cast mingling at the club.
Just like a guy and a girl pairing up to start a relationship, pairs of homologous chromosomes line up on the metaphase plate with one chromosome in each pair facing the poles during metaphase 1.
Like a relationship gone bad, in anaphase 1, chromatids separate, allowing the chromosomes to move. Chromosomes still contain sister chromosomes. The chromosomes move to opposite sides of the cell while being guided by the spindles.
In telophase 1, each half of the cell has a complete haploid set of replicated chromosomes made of 2 sister chromatids. These chromatids include regions that are non-sister chromatid DNA due to the crossing over from prophase. Cytokinesis occurs simultaneously, forming 2 haploid daughter cells.
The difference between meiosis 1 and meiosis 2 is that meiosis 1 separates homologous chromosomes, producing 2 haploid cells. Crossing over occurs in meiosis 1, which causes genetic variability. Meiosis 2 is similar to mitosis. The end result is the production of 4 haploid cells from 2 haploid cells.
Just like another trashy relationship with new partners, the second stage of meiosis is quite similar to the stages of mitosis. In prophase 2, the nucleoli and nuclear membrane disappear.
In metaphase 2, the chromosomes line up on the metaphase plate.
In anaphase 2, the sister chromatids move towards the opposite poles as individual chromosomes.
In telophase 2, the nuclei forms. Chromosomes decondense, and separation of the cytoplasm occurs during cytokinesis 2.
By the end of this process, there would be 4 daughter cells, each with a haploid set of chromosomes, all genetically different from each other. The importance of this process is that without meiosis, there would be no more genetic variability between parents and offspring, and there would be no evolution between generations.
When comparing mitosis versus meiosis, first of all, mitosis creates 2 identical diploid daughter cells. These are used for cell division and normal growth. For meiosis, there is created 4 unique haploid daughter cells, and these are used for sexual reproduction.
When comparing Romeo and Juliet to Jersey Shore, there is no real comparison.